Good morning, everyone, and welcome to Wave's 2025 Research Day, Spotlight on RNA Editing and RNAi. The slides that accompany today's presentation will be available following the call in the Investor section of our website at www.wavelifesciences.com. Before we begin, I'd like to remind you that management may make forward-looking statements during today's presentation. These statements are subject to risks and uncertainties that could cause our actual results to differ materially from those described in these forward-looking statements. The factors that could cause actual results to differ are discussed in our SEC filings, including our most recent annual report on Form 10-K and our most recent quarterly report on Form 10-Q. We undertake no obligation to update or revise any forward-looking statement for any reason. Today, we have an exciting agenda to cover, and our speakers will include Paul Bono, President and CEO, Chris Wright, Chief Medical Officer, Eric Engelson, Chief Scientific Officer, and Chandra Varghese, Chief Technology Officer. Following the presentations, all presenters will be available for Q&A. With that, let's get started. I'll turn the call over to Paul.
Thanks, Kate. Good morning, and thank you for joining us today. Our call this morning marks our 7th annual Research Day. We dedicate this time each year to showcase our emerging pipeline and novel platform innovations, and today we'll speak to how they're translating in the clinic. Ultimately, all of the updates move us closer to our vision for Wave, to deliver high-impact medicines and reimagine possible for oligonucleotide therapeutics and human health. For over a decade, we've been committed to innovation, leveraging our best-in-class chemistry and genetic insights to build a leading genetic medicines company. By combining human genetics, clinically validated chemistry, and novel biology, we have built a multimodal pipeline of RNA medicines that continues to translate in the clinic. At Wave, we are leveraging AI from target discovery all the way through our development process to add efficiencies and unlock new targets to reach more diseases with our different modalities. Today, we are spotlighting both emerging and clinical programs from 2 modalities, RNA editing and siRNA. In RNA editing, we are pioneering this field to continue to make history, most recently with our positive clinical data update from our ongoing Restoration 2 trial. In September, we shared exciting translation of RNA editing with WVE-006 from the first 2 cohorts of our Restoration clinical trial. These data included successful restoration of physiological or dynamic production of AAT protein at levels needed to prevent lung damage during an acute infection and represented a significant milestone for oligonucleotides. Building on this clinical success, today we will introduce WVE-008, our PNPLA3 aimer for liver disease. We are on track to submit a CTA filing for WVE-008 next year. In RNAi, our first siRNA, WVE-007 for obesity, is rapidly progressing as a novel approach grounded in human genetics that is designed to drive fat loss and preserve muscle. As we had previously shared in our INLIGHT clinical trial, the 75 milligram subtherapeutic cohort had substantial target knockdown, supporting our preclinical modeling that the second 240 milligram cohort would be expected to deliver a therapeutically relevant knockdown. To confirm this, we plan to analyze all activin E biomarker data as well as safety once the full expanded cohort 2 reach day 29. Today, we are excited to share an activin E target engagement update from the INLIGHT clinical trial. In the first 3 cohorts, we have observed highly significant dose-dependent activin E reductions following a single dose of WVE-007. These reductions in the clinic exceeded the activin E reductions that led to weight loss in our preclinical studies. The durability of effect supports the potential for once or twice a year dosing. We are incredibly excited about this program and its potential to disrupt the treatment paradigm for obesity by delivering fat loss while preserving muscle. Today's strong target engagement data sets us up for several clinical data updates with WVE-007 beginning later this quarter and throughout the first 2 quarters of 2026. We'll then take you through the latest innovations on our platform. With our novel chemistry, we are advancing our best-in-class siRNA, which builds on successful clinical translation of 007, as well as advancing our extrahepatic delivery capabilities to achieve both siRNA silencing and RNA editing. We are also using our platform to innovate new modalities, including the capability to simultaneously silence and edit two unique targets with a single oligonucleotide structure. We are poised for significant sustained growth, and based on our recent experience developing WVE-007, our GalNAc siRNA, it's clear that we can rapidly accelerate the time from target identification to clinical translation. Across our entire portfolio, we're advancing a diverse, sustainable pipeline grounded in genetic insights with the potential to treat well over 100 million people in the US and Europe. I will now turn the presentation to Chris for an update on WVE-006 in AATD. Chris.
Thanks, Paul. There are approximately 200,000 people in the US and Europe living with PiZZ AATD, which is often misdiagnosed and progresses over time due to accumulated tissue injury from unchecked inflammation. While it is one disease, alpha-1 antitrypsin deficiency impacts multiple organ systems with a predilection to impact lung and liver. AATD is caused by inheritance of a mutation in the SERPIN-A1 gene, which produces the protein alpha-1 antitrypsin. This AAT protein protects the lung during inflammatory or infectious events, preventing lung injury that could lead to emphysema and airway damage or bronchiectasis. Damage in the lung occurs during exacerbations that induce an inflammatory acute phase response. Individuals who do not suffer from AATD produce increased levels of protective functional AAT protein during these events, thereby preventing lung damage. In the liver, mutant AAT protein, also called ZAAT, tends to aggregate into deposits, which cause liver cell dysfunction and death, resulting in fibrosis, cirrhosis, and liver cancer. While therapeutics are advancing in development, the approved treatment options for AATD today are limited. There's an immense new need for more effective and convenient medicines. Weekly IV augmentation therapy is the current standard of care. This approach targets lung disease, providing healthy circulating protein, but it does not significantly impact liver disease, as the mutant ZAAT protein remains in the liver. In addition, IV augmentation therapy does not provide the dynamic increases of AAT expression needed during an acute phase response, resulting from a lung infection or other acute inflammatory events. In those with one disease allele or healthy individuals, the AAT levels will increase to meet the need at the time of an infection. However, with IV augmentation therapy, there's no way to go but down, unless an additional IV dose of protein is delivered to the patient. WVE-006 is a first and potential best-in-class treatment, which aims to address both lung and liver manifestations of AATD. 006 is an RNA editing GalNAc oligonucleotide that contains Wave's proprietary chemistry, including PN and N3 uridine. It edits mRNA A to I in a highly specific manner to produce only wild-type MAAT without bystander edits, and is administered by a convenient subcutaneous injection with potential for monthly or less frequent dosing. The goal of RNA editing is to increase wild-type MAAT protein at its source and restore the physiological production of AAT needed to prevent lung damage during acute inflammatory insults, while enhancing liver health by reducing the production and aggregation of ZAAT, thereby addressing both key disease manifestations. With RNA editing, you create a wild-type transcript, which leads to the production of MAAT, the healthy AAT protein in the liver. MAAT then circulates to reach and protect the lung. Its production in the liver also replaces ZAAT and allows for ZAAT reduction and clearance of harmful protein aggregates in the liver. Across genotypes, those with two wild-type alleles have normal liver and lung disease risk, and they respond to acute events by increasing their AAT levels to prevent inflammation-related tissue damage. Those with two Z disease alleles have high risk of liver and lung disease and are known to not increase AAT in the context of an acute phase response. The MZ patients with one healthy M allele and one Z allele have low liver and lung disease risk and are able to increase their AAT levels in the context of an acute phase response. Since the approval of weekly IV augmentation therapies to treat lung disease, the field is focused on keeping serum AAT levels above a minimum threshold of 11 micromolar, in part because ZZ individuals do not produce any MAAT and have a limited ability to increase serum AAT levels during an acute phase response or exacerbation. However, with RNA editing, if you edit by at least 50%, keep AAT protein above 11 micromolar, and importantly, restore the acute phase response, the risk for liver and lung disease should be low, as this would recapitulate the MZ phenotype and overcome key limitations of protein replacement therapy. Typically in the disease state, in patients with 2 mutant Z alleles, expression does not increase in the context of lung inflammation and the acute phase response. C-reactive peptide, or CRP, a measure of the acute phase response, can be highly elevated in relation to an inflammatory insult, such as pneumonia, but AAT does not correspondingly increase. In this context, inflammation remains unchecked and may lead to lung damage, causing emphysema and bronchiectasis. In the case of MZ or MM individuals that have a preserved acute phase response, AAT protein increases correspondingly to protect the lung against the undesirable consequences of inflammation, ensuring the lungs stay healthy. With our recent data from Restoration 2, it was very encouraging that we already achieved the key goals of RNA editing by restoring the MZ phenotype, even at the lowest single dose tested. Restoration of the MZ phenotype requires achieving 3 criteria. 1, keeping basal protein levels at or above 11 micromolar. 2, driving 50% or greater circulating MAAT with corresponding decreases in ZAAT protein. And most importantly, 3, restoring the physiologic response of serum AAT protein to acute inflammatory events. In our September data readout, we observed AAT levels up to almost 13 micromolar. We showed 64% of AAT was wild-type MAAT, while there was a corresponding 60% decrease in the mutant Z protein. These effects were highly consistent across individuals and persisted for up to 2 months after the last dose, supporting infrequent dosing of monthly or less. Notably, we were able to restore a ZZ patient's ability to respond to an acute inflammatory event, in this case, a kidney stone. The participant exhibited a strong AAT response of greater than 20 micromolar. The dynamic effect was observed 2 weeks after a single dose of 006, supporting the rapid onset of the editing effect. Encouragingly, the magnitude and the 4-week duration of this response were also proportional to the levels you'd anticipate in an MZ patient based on natural history. Following our data last month, we've had multiple interactions with key opinion leaders in the field who've expressed their excitement about these data, as the ability of WVE-006 to restore physiologic AAT production represents a major paradigm shift from protein replacement therapies. IV augmentation therapy provides a bolus of AAT with the intention of keeping protein above the potentially therapeutic threshold until the next dose. If a patient experiences an acute inflammatory event, the circulating AAT is consumed without replenishment by normal physiology. An interim dosing may be needed to protect against lung damage. By contrast, with 006 RNA editing, the levels of AAT can increase and remain elevated to meet the need of the body to protect the lungs, potentially protecting lung tissue from inflammation-mediated damage in a dynamic fashion, consistent with the natural physiology of AAT and the acute phase response. Additionally, augmentation therapies do not have any impact on the liver manifestations of the disease, whereas 006 has demonstrated the ability to meaningfully decrease levels of ZAAT protein. As we look ahead to the remainder of the Restoration 2 study, we're highly encouraged by our initial results and excited to advance the program, which is paradigm shifting and provides a potential new medicine to patients. With this goal in mind, we've made rapid progress through our Restoration 2 trial, We continue to dose patients in the 400 milligram MAD cohort and remain on track to deliver data in the first quarter of 2026. We've also initiated the single dose portion of our third and final cohort. Based on the continued favorable safety profile to date and the desire to optimize our assessment of efficacy, as well as dosing interval, we've selected 600 milligrams as our dose for this third cohort. 600 milligrams SAD and MAD data are expected in 2026. In tandem with our progress on Restoration 2, we continue to have discussions with our partner GSK about next steps for the program and planned regulatory interaction. With that, I'd like to turn the floor to Eric to review our next RNA editing program, PNP-LA3.
Thank you, Chris. Today, we're pleased to unveil WVE-008, our next wholly owned clinical candidate, which is a GalNAc-conjugated RNA editing program for genetically defined liver disease. 008 builds directly on learnings from WVE-006. As you heard from Chris, we've demonstrated clinically efficient and consistent RNA editing, restoration of dynamic physiological response to stimuli, durable effects supported in frequent dosing, and a favorable safety and tolerability profile. PLMP3 is a compelling target with strong human genetics evidence and a clear translational path to early clinical proof of concept. There are an estimated 9 million homozygous I148M carriers with liver disease across the US and Europe who are at a 9-fold higher risk of dying from the liver disease compared to non-carriers. As I will go over in the next few slides, we believe that our disease-modifying RNA editing approach to correct this variant back to wild-type function is going to be superior, not only to PLMP3 silencing, but also to generic non-precision medicine approaches in MASH. The PLMP3 I148M variant leads to a substantially higher risk of a range of liver diseases, from MAFLD, MASH, and alcoholic steatohepatitis to cirrhosis, liver cancer, and liver failure. Longitudinal data shows that heterozygous carriers have about 80% lower risk of liver-related death compared to homozygous carriers. Therefore, our therapeutic goal is to edit at least 50% of PLMP3 transcripts and hepatocytes, restoring a functional heterozygous state that is associated with a dramatically lower risk of liver complications and death. Given the large addressable population and a clear genetic linkage, this is a setting where RNA editing is ideally suited to be both precise and disease-modifies. While MASH among non-carriers of the PLMP3 I148M variant tends to be multifactorial and polygenetic, to a large extent driven by obesity and type 2 diabetes, I148M homozygotes represent a distinct subset of MASH with more severe disease biology with fast progression to end-stage liver disease. They're substantially enriched among lean MASH patients, that is, they're more frequently of normal weight, and they more often present with an advanced fibrosis. Up to 25% of MASH patients are homozygous for I148M, with even higher representation in lean and later stage disease. The only treatment options are non-precision medicines for obesity-related MASH, aimed at steatosis and early fibrosis with limited efficacy for these I148M carriers. Importantly, there are no approved genotype-directed disease-modifying therapies for PLMP3 I148M homozygotes. 008 is designed to directly address the genetic driver in these patients. PLMP3 has an important role in the regulation of triglyceride storage and secretion, supporting the formation of triglyceride-rich VLDL particles that supply lipids to the peripheral tissues. PLMP3 expression on lipid droplets is dynamic. It increases in response to feeding and participates in lipid remodeling and mobilization to meet cellular energy and structural needs. Additionally, PLMP3 has an important role in retinal metabolism by hydrolyzing retinal esters to release retinol. In short, PLMP3 is part of the liver's lipid metabolism machinery that determines whether fat is stored in the liver or mobilized and exported, as well as in retinal metabolism, disruptions of which can lead to inflammation and fibrosis. The I148M mutation impairs PLMP3's lipase activity and exerts a gain-of-function effect that inhibits ATGL-mediated lipolysis. This promotes hepatic triglyceride accumulation and stellate cell activation. Additionally, PLMP3 I148M suppresses retinol metabolism in hepatocytes and stellate cells, which increases liver fibrosis. Silencing PLMP3 can partially address disease biology, but is likely to leave residual pathology. ATGL activity is only partly rescued, and silencing will not restore retinal metabolism. As a result, silencing can partially address steatosis, but inflammation, ballooning, and fibrosis remain unaddressed. By contrast, correcting I148M is expected to restore PLMP3 activity and lipid mobilization, reverse steatosis, as well as inflammation, ballooning, and fibrosis. And this is the key rationale for RNA editing over knockdown, and would explain why PLMP3 siRNAs seem to have modest effect on steatosis, but no effect on later stages of MASH. In line with this, multiple preclinical studies highlight potential liabilities of silencing PLMP3. In iPSC-derived human liver organoids, PLMP3 silencing worsens steatosis. In primary human hepatocytes, silencing increases ballooning. And in stellate cells, PLMP3 knockdown worsens fibrotic responses. Together, these findings support our strategy to restore PLMP3 function with RNA editing rather than removing, aiming to improve steatosis, inflammation, ballooning, and fibrosis by preserving physiological roles important for liver health. WVE-008 is a GalNAc-conjugated RNA editor designed for subcutaneous administration with potential for frequent dosing. It incorporates Wave's proprietary chemistry and builds on learnings from WVE-006. On the left, you can see a dose response curve showing robust editing with 008. In the middle, RNA-seq results from 2 donors at different doses yielded an ideal editing profile. Strong on-target editing with no bystander edits and no off-target signals, as evidenced by lack of any other transcripts than the main PLMP3 transcript, giving us confidence in editing efficiency and specificity of 008. And on the right, we demonstrate high liver tissue exposure with 008 in preclinical studies, supporting the feasibility of a durable infrequent dosing. Collectively, these data provide a strong translational foundation, building on our clinical success with 008 as we advance towards the clinic with 008. Here, we compare lipid accumulation after treatment with our PLMP3 AIMer versus a PLMP3 clinical reference siRNA in 2 different in vitro models. On the top, in the HepatoPac, a 3D human liver cell culture model, we observed significant decreases of lipid accumulation after editing, while the siRNA did not show a significant effect. On the bottom, in a monolayer culture of primary hepatocytes, we again see substantial decrease of lipid accumulation increasing over time with our PLMP3 AIMer. In this model, we also see modest decreases with the PLMP3 siRNA. Taken together with prior literature, it shows the biological importance of keeping wild-type PLMP3 function and reinforces the biological rationale for correcting I148M rather than silencing PLMP3. In summary, 008 meets our criteria for proceeding into clinical development. Strong evidence from human genetics, a large patient population with no treatment options, the best and first-in-class opportunity, and an efficient path to clinical proof of concept. The PLMP3 I148M variant is a well-established driver of steatosis, inflammation, ballooning, and fibrosis. Yet, there are no approved medicines that directly address this biology. Emerging preclinical and clinical data indicate that simply knocking down PLMP3 is not the right solution. Loss of PLMP3 function can worsen the very features we're trying to treat. Our approach is different. With 008, we'll edit, not silence PLMP3, which will restore its important functions in liver lipid homeostasis. The goal is to reverse steatosis, inflammation, ballooning, and fibrosis by correcting the causal variant. 008 leverages Wave's proprietary, clinically validated RNA editing platform, giving us an efficient path to proof of concept in a large genetically defined population with no current options. Preclinically, our PLMP3 editing restores functional PLMP3 and decreases lipid uptake. And we have now selected 008 as our development candidate using Wave's proven chemistry and AIMer design. Clinical planning is underway for a first-in-human study where we will leverage previously genotyped populations to efficiently identify homozygous I148M carriers. In an initial first-in-human study, we will enroll homozygous carriers and assess safety, tolerability, pharmacokinetics, and pharmacodynamic endpoints aligned to the mechanisms of PLMP3 I148M. Our objective is a clear, early go, no-go readout for a precision medicine approach in this large population at high genetic risk. If successful, 008 has the potential to be a first and best-in-class treatment for homozygous PLMP3 I148M carriers with liver disease. We're planning for CTA submission in 2026. 006 and 008 are two clinical RNA editing programs, both targeting liver diseases, but our AIMer platform is broader. By building on our proprietary AIMer chemistry and design, we can modify our chemistry to see substantial RNA editing across a range of extrahepatic tissues. As we shared last year's research day, our AIMers achieve substantial editing in lung, heart, adipose, and pancreas, all without any ligands, as well as region-wide editing in CNS after ICV or intrathecal dosing in rodents or non-human primates. We've continued to make progress on hepatic and extrahepatic editing, and we're currently building a diversified RNA editing pipeline, including a range of indications, while advancing 008 towards the clinic. Now, I'm very excited to transition to our obesity program, WVE-007, a GalNAc siRNA that targets inhibin E. 007 offers a novel, long-acting, muscle-sparing approach to treat obesity and its metabolic complications. Individuals living with obesity face markedly higher risk of a range of serious conditions, including heart disease, type 2 diabetes, and several cancers. GLP-1 receptor agonists have transformed obesity care. However, their impact is often limited by a loss of muscle mass, tolerability challenges, especially GI side effects, frequent dosing, and high discontinuation rates. Beyond GLP-1 agonists, other treatments that enable fat loss have been shown to curb metabolic diseases. There are still large unmet needs. Our approach is to focus on healthy weight loss with substantial loss of fat, in particular visceral fat, which is a type of fat that contributes to the development of type 2 diabetes and cardiovascular disease. We believe 007 can address important gaps in the current therapy for the more than 1 billion people living with obesity globally. Our inhibin E program has a strong foundation in human genetics, which has been shown to increase the probability of successful drug development by up to 2 to 4-fold, with coding variant evidence in the upper part of that range. In the UK Biobank and other cohorts, heterozygous inhibin E loss of function carriers exhibit a healthier metabolic profile with lower abdominal obesity by a waist-to-hip ratio and lower visceral adipose volume, lower triglycerides, ApoB, and fasting glucose, and higher HDL cholesterol. They also have favorable associations with liver traits such as ALT and CT1, a measure of liver inflammation and fibrosis, and lower risk of type 2 diabetes and coronary heart disease. Our therapeutic hypothesis is straightforward. By silencing inhibin E mRNA by at least 50%, we aim to recapitulate the protective phenotype seen in these heterozygous loss-of-function carriers. Associations of target engagement biomarkers with outcomes have also been shown to increase the probability of successful drug development. Therefore, we're very encouraged by evidence from multiple data sets showing that higher hepatic inhibin E expression is associated with greater adiposity, insulin resistance, and MAPLD risk. Taken together, these data support that reducing inhibin E mRNA should drive healthy weight loss and improve metabolic health. Inhibin E, or inhibin subunit beta E, which is the full name, is produced in the liver, where two of the subunits dimerize to form the hepatokine activin E. Activin E gets released into circulation, where it binds in a specific manner to ALK7 receptors on adipocytes. Resulting signaling blocks adipose lipolysis, promoting abdominal adiposity and increasing risk for cardiovascular disease and type 2 diabetes. By reducing hepatic inhibin E mRNA with a GalNAc siRNA, we lower circulating activin E, decrease ALK7 signaling in adipose tissue, and release the brake on lipolysis, which is expected to shrink adipocytes and reduce abdominal adiposity, thereby lowering cardiometabolic risk. 007 uses Wave's best-in-class siRNA format and incorporates backbone stereochemistry and PN chemistry designed to enhance interactions with AGO2 and to improve silencing potency and durability. We have demonstrated the dramatic improvement of AGO2 loading, which is a crucial differentiator for us when we're trying to silence inhibin E, a target that is hard to keep sufficiently and durably suppressed, presumably due to evolutionary pressure for the need to store energy efficiently. A bit later, Chandra will go deeper on our siRNA platform and how it underpins our broader hepatic and extrahepatic pipeline. As we have shared previously, our preclinical data strongly supports 007's ability to both potently and durably knock down inhibin E, leading to impressive reductions in body weight and visceral fat while sparing muscle. In diet-induced obesity or DIO mice, a single dose of inhibin E GalNAc siRNA produces clear dose-dependent weight loss. Importantly, the weight loss is all driven by fat reduction, especially visceral fat, without any loss of muscle mass. As observed in the middle and the right panels, visceral fat was reduced by 23% at 3 mg per kg and 40% at 10 mg per kg, while muscle was unchanged. These data support 007 as a single agent option for healthy weight loss with visceral fat reduction and lean mass preservation. We also have robust preclinical data to support synergistic use cases with GLP-1 agonists. At a monotherapy, inhibin E siRNA achieves a similar magnitude of weight loss to semaglutide, but all coming from fat loss. When added on to semaglutide, we observe approximately twofold greater weight loss versus semaglutide alone. In the right panel, you can see that when GLP-1 treatment is discontinued, inhibin E siRNA curtails typical weight regain seen in controls, supporting its use as an off-ramp and maintenance therapy. In our DIO mouse models, a single inhibin E siRNA dose achieves more than 70% reduction of circulating activin E 1 month after dosing, at dose levels where we observed the substantial fat loss as I outlined in the previous few slides. When comparing the kinetics, weight loss with inhibin E siRNA was similar in magnitude to semaglutide, but occurred more gradually. Based on these data, we would expect a similar degree of activin E reduction to translate to healthy weight loss in clinic over time. In addition to weight loss, we expect meaningful improvements in cardiometabolic health, which is ultimately the main objective of any obesity medication. By reducing inhibin E mRNA and activin E levels, we would anticipate increases in adipocyte lipolysis and reduction of adipocyte size. This, in turn, would lead to fewer pro-inflammatory macrophages and less fibrosis in visceral adipose tissue, as well as improved insulin sensitivity, changes that can contribute to lower risk of cardiovascular disease and type 2 diabetes. On the next few slides, I'll show data demonstrating what we're seeing with inhibin E siRNA treatment, supporting the link of increased lipolysis with better cardiometabolic health. RNA sequencing from subcutaneous adipose tissue shows upregulation of genes and pathways related to insulin sensitivity, fatty acid utilization, and beiging of white adipose tissue. Concurrently, pathways involved in adipose remodeling and fibrosis are downregulated. These changes indicate better cellular energetics with suppression of fibrotic remodeling. As we can see, inhibin E siRNA leads to an upregulation of genes supporting better insulin sensitivity, fatty acid utilization, and beiging of white adipose tissue, while downregulating adipose fibrotic pathways. In visceral adipose tissue, we observe upregulation of glucose utilization, thermogenesis, and lipid metabolism pathways, consistent with what we observed in subcutaneous fats, and downregulating of innate immunity, cytokine release pathways, and extracellular matrix remodeling. Together, these findings suggest increased glucose and fatty acid utilization, reduced inflammation, and fibrosis in adipose tissue. As we presented at the ADA scientific sessions in June, histology confirms that a single inhibin E GalNAc siRNA dose also leads to adipocyte shrinkage in the DIO mice, with a significant reduction in mean adipocyte diameter. Additionally, histology demonstrates a shift towards less inflammatory state in visceral fat. Total macrophage staining decreases with a strong suppression of pro-inflammatory M1 macrophages, while anti-inflammatory M2 macrophages are maintained at the same level. Overall, this supports a transition from a pro to an anti-inflammatory state after inhibin E silencing. Consistent with these immune cell changes, we see a 53% reduction of adipose fibrosis at day 56 after a single dose of inhibin E siRNA, as shown by trichrome staining and quantified by image analysis. This decrease in fibrosis is likely the result of the shift towards a more anti-inflammatory state and will contribute to the improved insulin sensitivity and better cardiometabolic health. To summarize, 007 has the potential to be a convenient therapeutic that drives healthy, sustainable weight loss while preserving muscle. Our approach is deeply rooted in human genetics, which strongly support that heterozygous loss-of-function carriers show less visceral fat and healthier metabolic profiles with lower risk of type 2 diabetes and cardiovascular disease. Mechanistically, inhibin E silencing increases adipocyte lipolysis, shrinks adipocytes, reduces pro-inflammatory macrophages, and and fibrosis, and improves insulin sensitivity, all hallmarks of better cardiometabolic health. We've also shown that our clinical GalNAc siRNA 007 has the potential to be a best-in-class approach. 007 incorporates proprietary chemistry, including stereochemistry and PN chemistry, which increases potency and durability of silencing. And preclinically, we've demonstrated that a single dose of inhibin E siRNA achieves potent, durable target engagement with more than 70% activin E reduction, with a semaglutide equivalent weight loss, but all coming from fat loss, particularly visceral fat, with no loss of muscle. Further, our preclinical data shows that our inhibin E siRNA can double the weight loss compared with GLP-1s alone, and that it can curtail weight regain after GLP-1 cessation, supporting add-on and maintenance use cases. I'm now very excited to hand it over to Chris for an update on our InLight clinical study.
Thank you, Eric. As a reminder, InLight is a placebo-controlled single and multiple ascending dose study, randomized 3-to-1 active to placebo, with potential to escalate up to 5 single and 3 multiple ascending dose cohorts. It's designed as a safety, tolerability, PK/PD study, enrolling base cohorts of 8 participants that can be expanded up to 32 subjects per arm. Participants are healthy individuals living with overweight, with key inclusion criteria of A1C less than 5.9 and BMI between 28 and 35. In addition to safety, tolerability, PK, and activin E levels, the study has exploratory endpoints of body weight, body composition, and biomarkers. Our InLight trial continues to progress rapidly through the single ascending dose portion of the study. InLight is currently ongoing at multiple trial sites, including in the US, as we recently opened an IND. We began testing WVE-007 at our lowest subtherapeutic dose cohort, 75 milligrams, in 8 participants. Then for subsequent cohorts, which are expected to be in a therapeutic range, we had the option to expand to 32 subjects as warranted by safety. With a favorable safety profile, we have fully expanded cohorts 2 and 3 to date and are currently expanding cohort 4 at a dose of 600 milligrams. Our independent data monitoring committee has also approved further escalation to a next higher dose in cohort 5. Today, I'm excited to share the target engagement data with you from cohorts 1, 2, and 3, which included doses of 75 milligrams, 240 milligrams, and 400 milligrams, respectively. These data include time points out to 6 months from our 75 milligram cohort, which includes 8 participants, 6 of whom received WVE-007 and 2 placebo. We also have 1-month follow-up data from our 240 milligram cohort, which includes 32 participants with 24 on 007, and 1 month of follow-up from our 400 milligram cohort, including 8 participants with 6 on 007. I'm also pleased to note that to date, 007 continues to be safe and well tolerated with no discontinuations. We are delighted to share the robust target engagement we have seen to date. In this chart, we show the results for activin E decreases out to 29 days across the 3 doses and placebo. The percentage reduction of activin E from baseline is shown on the y-axis, with days on study on the x-axis. We observed no change from baseline to day 29 for the placebo group. However, we observed highly significant dose-dependent decreases from baseline across all 3 doses, starting as early as day 8 all the way through to day 29. Each active dose also showed statistically significant activin E reductions compared to placebo at all time points from day 8 to day 29. Across the 3 cohorts at day 29 compared to baseline, we observed a 56% reduction for 75 milligrams, a 75% reduction for 240 milligrams, and an 85% reduction for 400 milligrams. Based on our preclinical data, a greater than 70% reduction is expected to be in the therapeutic range for fat loss. And these levels were achieved in both the 240 milligram and 400 milligram cohorts. In addition to the highly significant and rapid dose-dependent activin E reductions we observed across our 3 cohorts, we had the opportunity to evaluate our initial 75 milligram subtherapeutic dose out to 6 months. I want to call your attention to this. Even out to 6 months, we continue to see some sustained reduction at this low dose. As you look towards the future data from our 240 and 400 milligram cohorts, which are ongoing, we anticipate that this durability could extend even further given the continued downward trajectory of activin E levels over the first 29 days. These data support a convenient dosing interval of once or twice a year. Our InLight trial data indicates that we have exceeded the reduction of activin E needed to show fat loss based on our preclinical studies. As Eric previously reviewed, and as you see on the left, we demonstrated that meaningful weight loss in the DIO mouse model occurred when activin E was durably reduced by 70% from baseline. It's important to note that we expect consistent activin E reduction over time is necessary to achieve weight loss in the same range as semaglutide. This level of reduction in our clinical data is highlighted in the green on the right-hand graph. This substantial decrease with a long duration of effect will allow us to assess weight loss across multiple time points. The InLight trial design allows evaluation of target engagement, blood-based biomarkers of metabolic health, body composition, and weight loss across multiple cohorts, with increased numbers of participants at higher doses as we expand each cohort. We look forward to following our cohorts as we progress with increasing doses and assessing these exploratory markers as well as weight loss over time. So what levels of weight loss do we expect to see in the InLight study? On the right are the semaglutide step 1 study results, where we separated the weight loss into fat in blue and lean mass in gray. From the fat mass loss perspective, which is most relevant to the inhibin E mechanism, there is a gradual weight loss over time with approximately 2.5% loss in fat mass by 3 months and 4.4% loss in fat mass around 5 months. The aim of the WVE-007 program is to observe fat loss competitive with weekly semaglutide by 6 months post single 007 dose. With the robust and durable activin E reductions and favorable safety we are observing to date, we're incredibly excited to continue investigating 007 in InLight and look forward to sharing multiple data sets, including target engagement, body composition, and weight loss over the coming quarters. Later this quarter, we expect to share the data from the 240 milligram cohort with 3 months of follow-up. In the first quarter of 2026, we'll have 6 months of follow-up data from the 240 milligram cohort, as well as 3 months of follow-up data from the 400 milligram cohort. Throughout 2026, we'll continue to have follow-up data for both these cohorts, and we'll also share data from the 600 milligram cohort. With a mechanism focused on fat loss with muscle preservation, favorable safety, and potential for once or twice yearly dosing, we believe WVE-007 has the potential to be a transformational approach for obesity, and we look forward to keeping you updated on our progress. Now, to share an update on our platform innovations, including our best-in-class siRNA, I'd like to turn the call over to Chandra.
Thank you, Chris. For over a decade, Wave has been extending the frontiers of RNA therapies through advances in nucleic acid chemistry. Our foundations began with novel and proprietary backbone chemistry, enabling us to apply principles of rational design to oligonucleotides and define structural activity relationship to single isomers. Since then, we have expanded our novel chemistry toolkit, which has provided step changes in potency, durability, delivery across hepatic and extrahepatic tissues. We have a clinically proven platform with unprecedented capabilities in silencing, splicing, and RNA editing. A hallmark of our platform is our ability to take shared learnings across modalities and apply these learnings to subsequent targets for rapid drug discovery and development. Today, we are pleased to introduce SPINA, a stereopure interfering nucleic acid design that enables RNAi-mediated silencing by further increasing AGO2 loading leading to improved potency and durability compared to our earlier siRNA designs and industry benchmarks. On the left panel, here you can see the dramatic difference in TTR mRNA silencing with GalNAc conjugated siRNA following a single dose at 2 mpk. All siRNAs have the same sequence and 2' modification as the literature reference construct with state-of-the-art siRNA chemistry. Compared to reference and our previous published siRNA construct, SPINA designs have substantially improved the potency and duration of silencing, and we have observed up to 95% TTR mRNA reduction at least up to 8 weeks after a single dose. We see similar differences in TTR protein and see unprecedented increase in potency and duration of activity with SPINA designs following a single 0.5 mg/kg dose of GalNAc siRNA. As a reminder, translation from preclinical experiments to the clinic is very well understood for RNAi, and we have just highlighted the exciting preclinical to clinical translation with WVE-007, our first SPINA design in the clinic with the potential for biannual or annual dosing. In our seminal NAR publication, we demonstrated that increase in potency and duration of activity compared to reference siRNAs was primarily driven by an AGO2 loading. However, the dramatic shift in potency and duration of activity of SPINA is driven by several fold increases in both AGO2 loading as well as PK, with SPINA driving up to 10-fold improvement in AGO2 loading versus reference. Now, I'll turn to some examples with our SPINA design to highlight how PN variants allows us to access new extrahepatic tissues, which in turn expands the scope of targets and indications amenable to RNA therapeutics. On this slide, we're showing results from a 3-month duration study with a single 5 mg/kg dose of 3 different tissue targeting SPINA variants in mouse experiments to silence SOD1 gene expression. On the far left, we are highlighting silencing in adipose tissue, and middle panel is silencing in the heart, and the far right is in skeletal muscle. In all 3 tissues, we observed approximately 75% knockdown after 1 month, and the level of knockdown persisted for at least 3 months following a single dose. Additionally, as you may recall from last year, we shared that with siRNA design, we are able to drive increases in potency and durability with tissue-specific delivery by optimizing physicochemical properties. On this slide, we're showing results from an 8-week mouse experiment using siRNA designs to silence gene expression. On the far left, we highlight the well-described impact of using GalNAc to access hepatocytes in the liver. But this also highlights the limits of a conjugate, and it is cell and tissue specific, so it does not enable silencing in other tissues of interest like white adipose. To the middle and the right, we show how we can alternate designs with PN variants to enable access to various combinations of liver and adipose tissues in the absence of any targeting ligand. Depending on the target and indication, we can deploy the designs that best fits the biology. Using PRISM, we can change the physicochemical properties of our oligos to deliver to numerous extrahepatic tissues and achieve potent and durable silencing with a single dose. Now, I would like to share with you an example of how we are applying these learnings and SPINA to develop first and potential best-in-class programs, which addresses areas of high unmet need. In this case, we have been working on targets with strong support by human genetics for a disease with high unmet need, high expression in liver and adipose, and measurable biomarkers, which offer efficient paths to proof of concept. In this slide, we show that we can achieve robust and consistent target mRNA knockdown and protein levels in the liver and in different adipose compartments utilizing both spina variants. Also, the knockdown of this target reduces fasting serum triglyceride by 60 to 70% using the two spina designs in a DIO model. Next, we set out to investigate cellular access and silencing in the kidney using spinas with PN variants. Oligonucleotide primarily accesses proximal tubule cells owing to its role in reabsorption. But this is usually a non-productive delivery, meaning that oligonucleotides do not modulate targets in those cells. Our initial studies with a single 10 mg/kg subcutaneous dose of our spina designs achieved a durable 50% knockdown in mice with 3 different SOD1 constructs, which persisted for at least 4 weeks. The panel on the right shows all 3 constructs had broad distribution, shown in red, across various kidney cell types. Now, to dig deeper into the cells that are accessible with spina and to confirm silencing activity, we collaborated with our partners at GSK to conduct single-cell RNA-seq analysis. This allowed us to map the knockdown effect at a cellular level, confirming broad knockdown across multiple cell types, including proximal tubules, distal tubules, endothelial cells, and podocyte cells. Building on these early successes, SAR studies helped to identify spina variants, which demonstrated an impressive 75% mRNA knockdown with the sustained 50% reduction lasting up to 3 months after a single dose of 5 or 10 mg/kg. Protein reduction was also confirmed by immunohistochemistry and miRNA scope staining for oligo-distribution. The knockdown was not only robust, but also widespread, affecting primarily the cortex and outer medulla regions of the kidney. High magnification images revealed broad distribution and substantial protein reduction, which was sustained at least up through 3 months following a single 5 mg/kg dose. Here we see decreased levels of light blue stain representing knockdown of SOD1 protein in treated samples relative to control. In red, we can also see broad cellular distribution of the oligos. In addition, we have demonstrated that through AIMer optimization, we can achieve RNA editing in kidney. Following a single dose of AIMer 1 and 2, we see that we can achieve 50% editing of UGP2 transcript with broad distribution. Finally, single-cell RNA-seq analysis was performed by GSK to assess editing efficiency. These data show efficient delivery and editing in multiple cell types in the kidney, including distal tubules, mesangial, podocytes, principal, proximal, and transient cells. Now, I would like to discuss an example of how we are applying learnings in chemistry optimization from across our platform to uncover new modalities. By using PRISM platform, we found a unique way to combine RNA editing and silencing modalities into a single oligonucleotide construct. This enabled us to silence one target while simultaneously cause editing or upregulating another distinct target with a single oligonucleotide construct. Additionally, because this oligo is loaded onto AGO2, we believe that this has the potential to further extend the durability of editing compared to an AIMer alone. We confirmed the ability of the single construct to engage in silencing and editing in vivo using a GalNAc conjugated oligo that is designed to edit UGP2 and silence TTR. As you can see in the graph on the left, in mouse liver, the single oligo construct shows a more durable level of editing, shown in pink line, compared to the UGP2 AIMer alone, shown in light blue line, out to 28 days following a single subcutaneous dose. As we just shared earlier, we believe this improved editing durability could be due to the loading onto AGO2. Furthermore, as the graph on the right shows, in the same mice, the single oligo construct completely knocks down TTR mRNA, just like the TTR SPINA alone. Since AIMers have the ability to upregulate protein expression by stabilizing mRNA, we explored the possibility of using a single oligo construct to upregulate one target and silence the other. As you may appreciate, simultaneously silencing PCSK9 and upregulating LDLR offers an attractive therapeutic approach to treat hypercholesterolemia. Using our single construct oligo, we were able to achieve exactly that. As shown on the left, using a single oligo construct, we achieved the same twofold LDLR protein upregulation as an LDLR AIMer alone. In the same experiment, the single construct oligo also silences PCSK9 to the same extent as PCSK9 siRNA alone. This shows how our approach can be used to upregulate and silence distinct targets using a single oligo. Beyond the examples I have shared with you today, we continue to push the boundaries of what is possible in the field of RNA medicines to unlock new targets and modalities with the ultimate goal of providing innovative therapies to patients in need. Now, for closing remarks, I would like to turn the call back over to Paul. Paul?
Wow, thanks, Chandra. Before turning to Q&A, I'll recap the updates you heard today and throughout the presentation. WVE-006 continues to advance in the Restoration-2 clinical trial, and as Chris shared, the single-dose portion of the third cohort is now underway at a dose of 600 milligrams, with the subsequent multi-dose portion to be dosed monthly. We're on track to deliver data from the 400 milligram dose cohort in the first quarter of 2026, and from the 600 milligram cohort, both single and multi-dose data in 2026. We've selected WVE-008 as our next RNA editing candidate for PNPLA3 liver disease, which will build on the successful clinical translation we've observed with 006. Turning to ENLIGHTEN, the highly significant dose-dependent activin E reductions observed post-single dose are incredibly exciting, not just for 007, but for our entire siRNA capability. The knockdown observed in the 240 and 400 milligram cohorts exceeded the levels observed in preclinical models that led to weight loss. Knockdown was incredibly durable and supports dosing of once or twice a year. Based on these data, we believe that WVE-007 has the potential to achieve fat loss on par with semaglutide by 6 months post-single dose, and we are on track to report clinical data updates, including body weight composition, starting this quarter. We also continue to innovate our platform and have robust extrahepatic delivery capability for both RNAi and RNA editing, and an emerging new modality that has the potential to unlock novel targets. Stepping back, our investment in RNA editing and RNAi to date has rapidly yielded multiple programs with WVE-006 and WVE-007 that have the potential to create immense value for both patients and shareholders. As you heard today, we are now expanding on the success of these programs and advancing the next wave to the clinic with PNPLA3, with more to come in both hepatic and extrahepatic. Finally, I would like to express our sincere gratitude to all the participants, study staff, and their families. They continue to inspire the work we do each day, and so from our entire team here at Wave, thank you. And with that, I'll turn the call over to the operator for Q&A.
Thank you. At this time, if you would like to ask a question, please click on the raise hand button, which can be found on the black bar at the bottom of your screen. When it is your turn, you will receive a message on your screen from the host allowing you to talk, and then you will hear your name called. Please accept, unmute your audio, and ask your question. We will wait 1 moment to allow the queue to form. The first question is from Joon Lee, MD, PhD, from Truist Securities. Please unmute yourself and begin with your question.
Hey guys, thanks for that impressive presentation. Your activin E target engagement, dose dependent, exceeding the DIO mouse model. Very impressive. How much more than 75% knockdown of activin E and DIO model did you need to achieve that semaglutide-like weight loss? And does the increase in fat burning through inhibin E drive increase in hunger drive due to caloric burning? And are you measuring potential increase in caloric intake of these patients? And what are you doing to control for that? And I have a follow-up on the platform, spin-up platform. Thank you.
Yeah, and thank you, June. I think it is important to note, hence the box shaded in green, as Chris shared, is that we are in the range from those DIO mouse studies. And so I think it's highly encouraging as we think, again, the 240 and 400 gives us a range to continue to suppress within there. So I think it's highly, again, encouraging that we have the levels of activin E reduction clinically that we need to see to set the clock. And I think just on that concept, and I'll turn it over to my colleagues, I think that's the most important take home from today, is that we've essentially set the clock. The essential durability that we've seen off of a subtherapeutic dose at 75 milligrams, where we see sustained knockdown out past 6 months. And as you saw in those slides, we continue to see that slope of reduction continue. And the fact that we can push that durability out even longer is highly encouraging. So in a lot of ways, the clock is set, and we expect this degree of knockdown to be sustained for a very long period of time, and therefore to be able to track together the level of weight loss that we can see relative to activin E reductions. I'll turn to Eric to have the discussion on the caloric intake.
Yeah, so we have tracked caloric intake in the mouse models, and we don't see a shift between inhibin E siRNA or the controls. What I did present today is that we actually do see some changes in some of the pathways in the fats directly, which indicates kind of an improvement of insulin sensitivity, fatty acid utilization, and beiging of fat. So there are changes to the metabolism, but we don't see anything in terms of caloric intake.
Got it.
I think that was actually going back to, that was, I think, one of the most impressive features of what I call kind of parity of caloric intake, is if we thought about that rebound weight gain. And I think that's an incredible opportunity as looking at inhibin E silencing as an off-ramp to GLP-1s, is that when we did the withdrawal study, actually the caloric consumption was mirrored in both mice. They both increased their caloric consumption on when removal of the downward pressure from the GLP-1 to actually go back and resume. And that hedonic eating, as many of you probably heard us refer to, that hedonic eating occurred across the control and the inhibin E treating. So even despite that pressure, we saw that we could, again, take the brake off of lipolysis and not store fat post the cessation. So again, highly encouraging as we think about a different profile for obesity treatments.
Yeah, and really appreciate that the weight loss is a little more gradual and takes a little longer than semaglutide. In mouse, it takes maybe 30 days longer. Extrapolating that to humans, any ideas as to how long we would need to wait to see that kind of weight loss in humans? And sorry to keep going, but I have a quick follow-up after that.
Sure, and I'm sure this is a question that's going to come up in the queue. So I think it's an important one. Again, we're on this journey together to understand inhibin E kinetics. I think we can feel highly confident based on the preclinical models that have translated with a positive control in the GLP-1s that these models do translate in the clinic. And so the effect of inhibin E reduction, activin E reduction, and weight loss in these models demonstrate that we do achieve those kinetic curves. And as you said, in preclinical models, it looks to be that there is a different slope or trajectory of that rate of decline. But we have a high degree of conviction that as you think about this 6-month time point, they do converge. And so this ability to watch that rate, we do think will come together. I think the question will be, what rate of decrease in fat do we see between the 3 and 6 months? And we'll have the opportunity to see that. And, as I said before, I think this is a target that, and there's probably many people dialed into the call that have extensive experience on the genetics, human genetics, biology, and have studied this target. I think what's incredibly compelling are 3 things. 1, we have the potency that's required, meaning we're in the therapeutic range of reduction of activin E. 2, as Chris said, and Eric, it is a combination not just of a single time point of reduction, but this feature of overcoming the natural compensatory mechanisms of pushing the target protein up are suppressed by duration. So we have that durability. And I think the third and most important thing in any obesity therapy, and really any therapy in general, is safe and well tolerated. And the fact that we can continue dose escalation gives us a high degree of conviction that the clock started. Medicine are in these patients. It's durable. We'll continue to follow it. But again, highly encouraging as we think about being able to continue to make these assessments over time.
Great. And then quickly on the SpinA platform, looking forward to your progress there. But how does it specifically for the muscle-tropic oligos? How does it come to transferrin receptor mediated delivery? Stop it. Thank you.
Andrew, you want to take that and follow up?
Yeah, this is yes. The duration, as you can see, what we showed here with the muscle. These are single small doses, which is very similar to a transferrin receptor antibody dosing too. So what we are seeing is, again, uptake. One thing you have to keep in mind is that it's good to deliver a molecule, but the molecule also needs to follow the mechanism. So we have an advantage here through our SpinA platform by increasing, not only delivering, but we're also increasing ago2 loading, which actually substantially increases the potency and durability. So that's.
I think just to follow up to Chandra's point, and maybe June, what you were thinking in the back of your mind and others do when they see muscle is like if you were to take kind of a DM1 like construct and imagine what you could see. I think the benefit to deliver muscle without having to use transferrin with the safety liabilities and the others that are associated with it, but actually not forgo the distribution, the durability, and in fact see potentially longer durability based on the siRNA constructs we have, I think open up tremendous opportunities. So I think as we think about the opportunity in muscle more broadly, we don't have to sacrifice what others are showing with a conjugate to deliver to the tissue, but we can get that potent, durable knockdown without that. And I think that's, again, highly encouraging as we think about the platform capability and future targets.
And maybe just to add one more point on that is that, just recall that our chemistry is compatible with the ligands when very useful, such as GalNAc. So, but we don't have to, it's not an either or. We can use it when it makes sense, but for transferrin to get to muscle, it doesn't make sense because we get there very efficiently. And we have shown that already with our DMD medicines.
Great update all around. Thank you so much.
Thank you, Jim.
The next question is from Joseph Schwartz at Leerink Partners. Please unmute yourself and begin with your question.
Great. Thanks so much for the presentation. I have a question on 07 and maybe one on 08 as well. So I guess given the early fat loss signals might be in a fairly low single-digit range after 3 months of dosing of 07, I was just wondering if we'll be able to see the slope of response in patients similar to what you've shown preclinically as well as, on the activin E biomarkers. That way we might be able to appreciate how the effect could evolve with more time.
Yeah, I mean, I think it's about the data, and as we laid out nicely, and Chris laid out a number of time points over a number of doses. I think we'll be able to plot those kinetics and what that slope of that trajectory begins to look like. I think as you get early, you remember that slide at 1 month, you're in that like 1.5% range. And we all can acknowledge that in smaller studies, a high degree of variability at early points in time. But I think these moments in time at 3 months, 6 months, and across doses will allow us to eventually, and remember the follow-up goes out for these therapeutic cohorts out to a year. So I think we're going to have the ability to really track what fat loss looks like with a completely novel approach to treating obesity that drives healthy, sustainable weight loss. So I think, again, highly encouraging as we think about this sign. But yes, by 3 months, we'll get a sense of where we are on that trajectory across both body composition, body weight, and other biomarkers.
Okay, great. Thanks. And then on 08, given 148M expression is enriched in lean MASH patients who present later, I wanted to ask about your diagnostic and enrollment strategy to enroll these patients early enough in the sequelae that 08 can help. And also, can you talk about the biomarkers which you can track at various stages of development to gauge the degree of target engagement at different doses? And what is your hypothesis about the dose levels that make sense to study in these patients?
I'll hand it to Eric. We're not sharing yet, although we have biomarker data, and as one would imagine, sometimes biomarker evaluation is competitive in this field, and we've made strides to, as Eric pointed out in this discussion, to derive biomarkers there. But I'll let Eric talk holistically about that.
Yeah, we haven't shared any details on this really, but there are good non-invasive biomarkers that we can apply early on. I think it's important, one thing, Joe, to point out is that the variant per se is associated with increased risk across the whole spectrum. So it's from F1 all the way up to F4. It's just that it's more enriched and kind of a big segment of individuals in the later stage MASH, and especially lean MASH, that don't really have any treatment options. But for a proof of concept, it's also possible to go to a more all weight loss, like all ranges of BMI, or early stages or late stages, and we haven't really shared exactly where we're going right now.
Okay.
Just to be clear on that. So it could treat all patients that have the mutation. And again, we're talking about MASH in this context, but as Eric also pointed out, there's a range of liver diseases associated with PLA3, and so thinking about it more broadly gives the opportunity for expansion to other indications in the future as well.
Great, thank you.
Please try and focus questions to one question and one follow-up. The next question is from Steve Seedhouse at Cantor Fitzgerald. Please unmute yourself and begin with your question.
Good morning. Thanks, everyone. Maybe just wanted to cover at the end there that bifunctional RNA editing approach. You just kind of slipped it in at the end there, but it looks awesome. The PCSK9 LDLR application in particular, that feels like not just a proof of concept experiment, but actually something that I would want to see in the clinic now. A pretty promising approach. So how close is that, in fact, to the clinic?
Well, we're not guiding yet today in the clinic, but as you point out, I think we have two independently really well-validated targets that should be developed. Obviously, PCSK9 extraordinarily well validated. And I think in the introduction and hearing lots of feedback when people are excited about what we could do with LDLR, there's always this discussion that goes, well, what about PCSK9 and how do And I think to the point that the team takes on is there's a lot we can learn about a platform. And I think if you take anything from today's call on a research day, that the research day is incredibly translational. When we spend time thinking about something that could be a new platform, in the case of these dual conjugates, it's not esoteric. It's not thinking about what can do, but actually, as you point out, how do we apply it to high impact, high value programs to medicines where hopefully the outcome is how quickly can we move this forward. So a lot of work is happening to accelerate those programs. We see lots of translation from research day going forward, and there's lots of good things behind this, as Chandra shared, in the kidney as well. But yes, these are two highly compelling targets, and you put them together, you get a very highly compelling program in cardiovascular disease. So we're excited to continue to make progress there.
Great. Just maybe more near-term question then. So you announced today, I guess, that you're dosing up in a couple of these studies. So AATD going to 600 milligrams, and then in the obesity study, 600 and beyond. It looks like, despite already obviously really good PD data. So can you just elaborate on those decisions? And it feels like the right call in AATD to try and maximize editing efficiency. But what are you seeing in the PK/PD data that inform those decisions?
Yeah, I think as Chris shared, we were modeling up to 600 because it's the last and final dose to really, max out where we are on the dosing curve. We can because of safety, and two, no efficacy gets left on the table. But most importantly, as you remember from the prior call, we see healthy levels of alpha-1 antitrypsin protein 2 weeks after a single dose of 200. So there's also the optimization of PK and what those dosing intervals like. Are they monthly? Are they quarterly? So to fully flush out the dosing interval, and obviously we're collaborating on this program, I think we'll learn a lot from 600 that ultimately enables us to say that portion of the study is complete, so the study can move on to the subsequent portions. As it relates to Inhibin E, I think it's, as Chris alluded to, highly encouraging that we can go above 600 and still have the DSMB allow us to go higher. So I think it's highly encouraging from a safety perspective to go higher. As you saw, we're doing a phenomenal job on activin E lowering. How much more is to be gained at going higher? But, again, we'll learn more about duration of activity as well as, continue to establish, profile for a medicine. But again, we're highly encouraging from just the 240 and 400, let alone the 600 and, where we can go from there.
Great, thanks so much.
The next question is from Yang Zong at Wedbush Securities. Please unmute yourself and begin with your question.
Hi, good morning. Thank you very much for taking the questions. And the first one is on the obesity program. I wanted to confirm that you're still going to report weight loss data from the 75 milligram dose cohort this month. And based on preclinical data, so given that the knockdown efficiency is still slightly below that 75 that you talked about, is it reasonable to still expect some kind of a weight loss signal? And if that's the case, well, how much difference will maybe a higher knockdown efficiency translate into weight loss?
Yeah, I think we have to be focused. 75 was modeled to be subtherapeutic because it's a phase 1 study still and needed to be dose low. I think to your point, highly encouraging to see durable, stable knockdown out at 6 months. So yeah, actually stepping back, short answer to your question is yes, we'll show weight loss data from the 75 milligram. And that was on Chris's slide at this Q4 time point will be 75 as well as the 240. We wouldn't be guiding this expecting to see something given the range was outside of the early reported data. But again, this is the first time Inhibin E has been developed in the clinic and will have long-term durable knockdown. So we'll get a good sense of that. I also look, whether or not we see other biomarkers that are interrogated beyond just weight loss itself, I think will be helpful. But again, it's a 75 milligram cohort. So it'll be, if anything, level setting for where we get to with 240 as that continues.
I see. And
that will be included.
Okay. Yeah, that's very helpful. Then on the 008 program, I think, well, is it reasonable to expect that initially you will be focused on MASH? And I think, do you have any data to show whether those carriers, they are showing any difference response to current MASH treatment or other therapeutic in clinical development? And there are other agents, for example, working on fat synthesis modulation or fat metabolism. Do you expect your approach, I know it's a precision approach, will potentially generate something different, maybe better efficacy when it narrows down to that specific patient population?
Yeah, thanks. That's a great question. Yeah, that is exactly the point of this. It's a disease-modifying correction of the causing mutation in these patients. And while you could kind of potentially have some effect of other medicines, is going to have lower efficacy than correcting the driving, disease-causing variant.
Okay, great. Thank you very much.
The next question is from Salim Syed at Mizuho Securities. Please unmute yourself and begin with your question.
Hey guys, thanks for the question and the color today. So Paul, Chris, I just want to, because I know a lot of people are looking at a quantitative number, and you kind of put one out today, but not really on this obesity stuff that we're getting in the 4Q here. Just looking at cohort 2, which is your therapeutic range, the 240 mg, where you do have 75% E reduction. That's kind of the target you stated here. The SEMA is showing 3 months on your chart a 2.5% fat loss. So quantitatively, is that the bogey here? And why should we not be looking at other things like amylin or even like the Nomura oral data? Why aren't those the relevant comps here? Thank you.
Yeah, I think we use SEMA because the GLP-1s have been the comp that we could use to model mouse weight loss and mouse fat loss to humans. So I think finding a harmonization of benchmarks that we can use sustainably across programs where we could look at ranges of fat loss and be able to take those ranges and extrapolate them in a forward-looking way. Again, this is healthy overweight volunteers as we move into subsequent studies, being able to try to create benchmarks on a new modality that let us look at ranges of fat loss, I think is important. I think to your point on where we are, I think the recognition is at the 3-month time point, you have this range of what we've seen preclinically, where it does look like in the early point, the kinetics on lipolysis versus what happens when you have chemical starvation and lose muscle and fat look different. And so I think our view has been that we do expect to be on that curve of weight loss and particularly fat loss, and then be able to see that normalize. And so I think by the time we get to 6 months, we think we're over that hurdle on the normalization. But it just means where we're going to be to try to put a pin in an exact moment of time relative to the GLP-1 at 3 months is something we're going to have to learn in running the experiment. I think in order to have the conviction to even run the experiment, you need to believe that you have activin E reductions that are within the range. And I think, as Chris alluded to today, we're clearly within the range. And so I think that's going to be helpful as we look at these subsequent experiments, both at the 240 lowest therapeutic dose as well as others, and continue to follow this over time. And I think by the middle of, as we move into 2026, as Chris shared too on the slide, we're going to have a number of patients with therapeutic ranges of activin E reduction that are highly consequential, and be able to look at not just where it is at 6 months, but continue to see, what that range of reduction looks like, both in terms of time and dose.
Okay, but we're getting 3 months, right? So I just want to be clear, because the consensus is all over the place. Like, investors are all putting out different numbers. I just want to be clear to get consensus in a little bit. Like, people should really be thinking low single digits for this 4Q update we're getting.
Absolutely. I think we were very clear on the numbers that we have put out, that even if you're in a GLP-1 for fat loss, again, we have to separate for other weight loss versus what we know about the degree of fat reduction. Inhibin E pathway, because it is, as Eric alluded to, working across what actually drives healthy weight loss, meaning preserving insulin sensitivity and all of the other cardiometabolic parameters of weight loss, is by reduction of fat and not muscle. And so when we look at that degree of fat loss, I think we were very clear about the range of numbers. And as you pointed out, low single digits is what you see with fat loss within the GLP-1 space at the early time points.
Okay,
and just to add to that, like the other things that you're bringing up, like amylin or any incretin medicine or any obesity medicine in development right now, they're all have an orthogonal, like, different approach. They're all kind of acting on appetite or GI, versus we are acting directly on the adipocyte. So it's a totally different approach, but the comps are always going to be like the fat loss and primarily the visceral fat, which is what increases your risk of cardiovascular disease type 2 diabetes.
Okay, thank you so much, guys.
The next question is from Cheng Li at Oppenheimer. Please unmute yourself and begin with the question.
Oh, hi. Thanks for hosting the event and taking the question. Maybe a two-part question from me, one on 07. I'm just wondering if you can comment on the patient baseline in the cohort 2, and just based on the patient baseline, whether the semaglutide from step 1 is a fair benchmark, and I have a follow-up on 08.
I'll let Chris comment on that.
So the question was about the patient baseline compared to SEMA?
Yep.
Yeah, so we're looking at patients that are healthy with overweight, and so as you mentioned, we mentioned the BMI range and the A1C, so they're non-diabetic and have an A1C range. I have a BMI range, I think that's up to 28.
Yeah, so I think the key is that they're similar in both the SAD programs across both, as you're pointing out to the step 1, and this would be similar.
Okay, that's helpful. And on 08, congrats on the preclinical data. It just seems like the RNA editing efficiency you show is around 60 to 70%. So I'm just wondering, is there like a ceiling for maximal RNA editing you can achieve, or there's something else you can further optimize by maybe increasing the loading to the ADAR enzymes, or any way to improve the RNA template design with potential learning from the siRNA modality? Thank you.
Sandra, do you want?
The RNA editing, so if you look at it, when you look at Sanger sequencing, which is the plot that we have on the left-hand side, that you're talking about at 75%. But when you look at the transcript, when we did the transcriptome-wide analysis, we see about 85% editing. So this is actually just the nature of the assay. So we're not saturated. It's very similar to what we observed with 006 as well.
Got it.
I think that's something, to Chandra's point, it's something that we learn now with human clinical data, right, from the prior study, and being able to look at what occurred during an acute phase event is, we learned that the drug is not substrate limited. There's plenty of drug. We had editing 2 months after the last single dose, the lowest dose. So there's plenty of drug on board, so it's not substrate limited by the stability of the construct. We also learned that when you have the acute phase event and increased transcriptional activity, the enzyme's not substrate limited. And so I think those are two highly important features as we think again about what we continue to do in optimization, is realizing that, we did prove that ADAR is a catalytic enzyme in there to be edited. And to Chandra's point, our medicines are highly stable and durable. And so again, it gives us a lot of work that we can continue to do in terms of driving further optimization, both in the liver and then importantly outside the liver.
Got it. Thanks, and congrats again on the update.
The next question is from Cha-Cha Yang at Jefferies. Please unmute yourself and begin with your question.
Hi, this is Cha-Cha on for Roger Song. Thank you so much for hosting this event and for taking our questions. I have two, both about your 007 program. So the first one has to do with some of the safety fronts. I know you said no discontinuations, but I'm hoping that you can give some color on the GIAE safety front. And then my second question just has to do with benchmarks as well to sema and tirzepatide. So I know you talked about benchmarking to visceral fat loss, but I'm wondering if you believe that the higher dose cohorts for 007 could achieve overall weight loss comparable to sema.
Yeah, I mean, I think we have to think about fat loss, weight loss, and I think we've got to stay focused on mechanistic activity, which is fat loss. But again, highly encouraging that DIO mouse models were achieving weight loss similarities. But again, we have to look at that as a model and mechanism. So to the point on could and potential and forward-looking, particularly across doses, the models don't suggest that it's inferior to. So I think that is always a possibility. But the focus has been on optimization of fat loss relative to the fat loss that's seen with GLP-1s, and that's what we'll continue to drive and explore. But again, higher doses, time, and the ability to recapitulate what we've seen in the animal models again are highly encouraging. As it relates to safety.
Yeah, so as we mentioned, we've been dose escalating without any issues from the safety perspective. It's been safe and well tolerated, and the DMCs have been allowing us to proceed without any questions. All of the AEs that we saw were mild in terms of any drug-related AEs, and given the mechanism, you wouldn't really expect any GI symptoms.
It is important to note when we think about the GI toxicities, and Eric brought this up in terms of a lot of the category classes. It's not an incretin. It's not working on either suppressing appetite or slowing the GI tract. And as just Chris just said, this is taking the brakes off of lipolysis. And again, highly encouraging that it's a single subcutaneous dose with long durability. So both on the preclinical studies as well as what we're seeing in the clinic. Again, it's got a profile that has us encouraged for a long, durable medicine. And I think the fact that DSMB could review the current data, let us to go to 600, see the current 600, and enable us to go higher, again, highly encouraging.
Wonderful, thank you.
Yeah.
The next question is from Catherine Novak at Jones Trading. Please unmute yourself and begin with your question.
Hi, good afternoon, everybody. Thanks for the great presentation. I have a question on 007. Can you let us know with the InLight study, what is blinded to the sponsor? Obviously you're sharing target engagement data while the study is ongoing. So is this unblinded separately and, safety weight loss is still blinded, or when a cohort finishes dosing, is anything unblinded at that time?
Yeah, I think stepping back, it's important to realize what I said at the very beginning of this call. So the short answer to your question is yes. We did an analysis here looking at activity. Obviously, DSMBs can look at data, both blinded data, and in preparation for those, and the DSMB can continue to make their decisions based on evaluation of those data sets. But no, I think it's really critical that this wasn't an unblinding of the totality of data sets, being able to even look at unblinded weight loss data, which I know some people are thinking. This was a analysis that, if you remember the last update, and I'll kind of go back to the beginning of what I wanted to reiterate, is a lot of the forward-looking pieces are predicated on being within a range where we've got a good model that enables us to predict where we're going to be. And when we could model and say at the 75 subtherapeutic, we had substantial target engagement. I know there were a lot of questions of what range of target engagement were you seeing? how do you know that, your 240 is going to be within a therapeutic range? And, we have a phenomenal team who's doing a lot of modeling. I think to see where we needed to be able to be on that trajectory to deliver weight loss data, we knew we needed to be within that range. And yes, we were modeling to be in that. So this was a very specific cut at day 29 to do that assessment and assure that this 240, where we said we should be in that range, we can now switch that for should be in the range to we are in the range where we see it.
And just to add, so we will be unblinding for our 3-month readouts and for our 6-month readouts with regards to weight and the entire study and other measures, such as body composition and biomarkers, but not for the big 9 measures. We just were looking at biomarker target engagement of activity.
Got it. And just one more, if we could give me any specifics about the timing of the data cut showing activin E knockdown, even though we have 24 patients in the 240 mg cohort at 1 month. Given that, we know enrollment was complete before the end of July, is there a reason you don't have 1 month data for all 32 patients in that cohort? And how long after the 3-month follow-up period do you anticipate it would take to analyze once the data are unblinded?
Yeah, I think we haven't guided beyond where we can get to, but each one of these cuts we were up and dosing, and that's rolling forward. So as we said, day 29 are the cuts of all patients who had that data at that time. So these were the patients at this time of the cut of the day 29, full 32 on 240 that pulled that data together. I can't guide, other than we'll have data this quarter on the 3-month follow-up of those patients, which will include then also the 75.
And just recall that the study is designed so that you have a cohort of 8 initially, and then if the safety and tolerability is acceptable, which has been in every case, you can expand the cohort, and all those subjects are not enrolled at the same time. So just maybe that gives you a sense of the flow of the study in terms of enrollment.
Okay, thanks. Thanks for answering that.
The next question is from Bill Morn, PhD from Clear Street. Please unmute yourself and begin with your question.
Good morning. Thanks for the question. So just on the Inhibin E program, can you talk about the key remaining unknowns in the biology, given this is kind of the first foray into lowering activin E? Whether there's a level of reduction that is too much, or whether long-term there may be some compensatory metabolic changes that may kind of offset some of the effect.
You presented, George. Sure.
Yeah, I think part of this is like relying on the human genetics. We know that on the on target, there are really no concerns about decreasing it, because they're also homozygote carriers of the loss of function carriers, and they're actually healthier. So with regard to that, and we also do pheno-wise studies where we look at these carriers across every known phenotype in human, and we don't see any increases of anything. So that gives us some reassurances. Then, we've obviously taken this through talks, and it's all, both on and off target is clean. So with regard to that, no really concerns about this mechanism with regard to decreasing it. I think we can decrease it, that, there's really no limit to how much we could potentially decrease it.
Okay, and the biology on the other side of inducing it, as you were saying, we have shown on the other side, as Eric shared, that, we do think that, we've shown across the pitch that, inducing it impacts a whole bunch of features, right? Beyond just fat loss, as Eric shared, when we think about other drivers of adipose inflammation and all of the features of what makes this, I think, really important, a cardiometabolic drug. I think we need to step back and think about the totality of it. Yes, it has an impact on fat loss and weight loss, and that's important. But if we think about obesity as truly a public health disease and metabolic, and why fat loss and muscle sparing is crucial, it's critical. I think that's ultimately what we think is going to be the biggest driving differentiator in this program, is for insulin sensitivity, is saving muscle. That is important.
Yeah, and I think this is a largely new target since just a few years, but even today with some of the data we've disclosed today, we're starting to put together the pieces how basically increase in lipolysis is contributing towards the lower risk of cardiometabolic disease seen in these genetic carriers of the homozygote or the heterozygote loss of function carriers. So I think the pieces we put together today with an increase in insulin sensitivity, a shift from the pro-inflammatory to the anti-inflammatory state in visceral fat and lower fibrosis, all of those things together contribute to the lower risk of cardiovascular disease and type 2 diabetes. To Paul's point, which is ultimately really the reason for any obesity treatments.
And on PNPLA3, is that a validated enough target that you expect to be able to put forth an argument for accelerated approval, or do you expect to have to show functional endpoints, and what might those be?
Let my colleagues start, but
I mean, it's a very well established target where the biology is well known. I don't think we would guide towards regulatory interactions at this point, but to the question, it is a very well known mechanism. Yes.
With biomarker, and to Eric's point that we made, I guess, on one of the prior questions, that we can have a biomarker driven response in terms of then targeting the genetics, the biology through biomarkers, then ultimately through phenotypic analysis of the patients.
Great, thank you.
The next question is from Madison El-Sadi, PhD from B. Riley Securities. Please unmute yourself and begin with your question.
Hey everyone, thanks for taking our question. I appreciate the update. Lots of things one can get excited about. Maybe first, given the MASH liver field pivot to these non-invasive tests, does this allow Wave to execute really end-to-end a phase 2 or even a future phase 3 in a 148 variant population just as an independent company?
I mean, I think we were excited when we saw the transition finally to looking at non-invasive imaging as endpoint. So I think to the prior question and to this one, I think it's what obviously elevated the target in our minds, that we've got a highly genetic correlation. I mean, it's in commercial genetic testing. So it's a readily accessible genetic biomarker that drives disease. Being able to modify that with a biomarker and non-invasive imaging is something that we can fully deliver.
Got it. And then secondly, Paul, has your view changed, I guess, on the future potential real-world use of 007? You've spoken about it in terms really independent of GLP-1s as a way to lower GLP-1 dose, to improve the GI tolerability, and also as an off-ramp. Just wondering, as more data has came out, as you've learned more, if your view here has changed recently.
No, I mean, I think on one hand, probably it's easier to have conviction with clinical data than preclinical data. So I think having seen the range of knockdown, the durability translate, you do a lot of modeling when you try to go from a DIO mouse to human. And I think the more data points we can have on actual protein reduction and durability of protein reduction, the more conviction you have. So I think it absolutely is going to be a very important cardiometabolic medicine. I think where commercially the uptake starts and whether or not people begin to use it in advance with the evolution of just GLP-1s and where they go, particularly around genericization. I think there's easily for fat loss, muscle sparing, cardiometabolic improvement. If the goal is to treat obesity, I think no doubt there's no wavering from our point that it can be very much a frontline medicine. Where it gets utilized in the initial piece, which I think is a very consequential market, is sustaining people on a GLP-1 where they're going to continue to lose bone and muscle over time. 70% of patients can't stay on it. The ability to transition, as we did on maintenance, where you can move patients to a twice a year sub-Q injection and know that you're not going to get weight gain, you're going to sustain that benefit over time, I think is a phenomenal use. I mean, for the US where GLP-1s are used, but we have to look broader than that. I mean, there's over 1 billion patients worldwide living with obesity. And so when we think about what those treatment paradigms look like, I know we often look at this particular market and say, well, how are we going to intersect there? But the ability of markets where people don't have access, I mean, you have large companies buying, manufacturing CDMOs to try to keep up with demand and just individual marketplaces, the ability to think about how you can scale a once a year sub-Q GalNAc siRNA opens up a massive possibility of how you think about the global treatment for obesity in a way that we believe this mechanism has the potential to address. And so highly encouraged as we go on, we actually are set to deliver, as I said, the clock started, medicines and patients, we're knocking down the target in these ranges, and we're going to be able to follow this out over time. And I think as we do that, I think that will be a huge opportunity as we think about the global obesity market. I think that's really how we think. How do we treat the over 1 billion patients worldwide living with obesity?
And just to add, I think we're really excited about all 3 use cases that we have presented, and we hear a lot of that sentiment when we're out speaking to KOLs and the community as well. That there's really an opportunity in all of those 3 kind of potential paths forward, and we're going to be guided by data to move forward now.
I'll bring up one other point, and I think that as Eric just alluded to, I mean, we spend a time, obviously our heritage has been in rare diseases, and we spend a lot of time with patient community. And as we bring patients in, I think carefully about the number of patients who tell us when they watch ads on television advertising the weight loss potential of GLP-1s, and they truly say, like, even if I achieve that level of weight loss, I still wouldn't be at a healthy goal in weight reduction. And so I think as we do think about what are the arrows in the quiver, as to Eric's point on talking to KOLs, that can actually enable patients to achieve true healthy weight loss that's going to drive protection for patients living with obesity, there very much is the need to think about a multitude of medicines that can be used together. And I think, as we said, the fact that we don't work on the GI system in terms of causing nausea, vomiting, distractibility, the fact that we don't cause anhedonia because we're not actually driving appetite suppression in the CNS, the fact that we've got a very orthogonal approach to doing this and can show that we have complete synergy with the incretins, so in this case GLP-1s, I think does allow us to think about that middle use case, which is really about how do patients continue to be able to achieve those level of weight loss if they need it. So again, I do think that this orthogonal approach gives us a huge opportunity to think about all categories of how this makes a difference.
Got it, understood. Really appreciate all the color.
Yeah, no, thank you for the question.
The next question is from Samantha Semenkow, PhD from Citi. Please unmute yourself and begin with your question.
Hi, thanks very much for taking the question, and congrats on all of the updates you've presented here today. I just have a follow-up on one of the prior questions for your RNA editors, and I guess specifically for 006. What levels of optimization could you look at to help increase the maximum editing threshold? And as you said, if that threshold is not substrate limited, is it then concentration dependent? And is that why we're seeing the editor respond when endogenous production of AAT increased in that one patient with the acute phase response? Thank you.
Yeah, no, thanks for the question. And, we'll tag team this. I think the interesting thing on 006 independently, I think that's some of what is intriguing when I think, and I know sometimes when I say this, I'll get nothing's easy in drug development, but, I think the difference between 006 and some of the other targets in editing, in some ways is 006 is one of those where you've got this other kind of variable, right? What happens during an acute phase response? So the other variability is increased transcription. That's what, as we shared again today, that's the biological rationale and mechanism for AATD is you get this response, this inflammatory response, CRP goes up, you get this increase in transcriptional regulation. And so there's a different texture to how that happens. This gets to that target differentiation. And, again, it's when we have targets like PNPLA3 and others, as Chandra shared, we've got other features in terms of stability of showing good, stable drug activity and editing. And we do see a high degree of reproducibility across constructs, which I think, again, there's 2 metrics that we look for in kind of saying, defining a platform is how much similarity is there to move from one construct to the other, and how do you impart those learnings? The other that I use a lot, is how quickly, when you have a platform, does it mean moving from a target to moving to the clinic? And, we use that also as a barometer of the efficiency with which there's portability. And I think, we've been highly successful in that portability concept. I mean, Inhibin E was 18 months from our target work on the in vivo data to being in the clinic. And so I think we've been able to impart a high degree of that. And, again, with PNPLA3, we see the similar. I don't know if others have
I mean,
additions to the.
maybe
Sorry.
the only thing to add is just, the way we're thinking about this acute phase response, and it's pretty well understood that you increase expression, at the RNA level. So if you have more RNA, there's enough, of the molecule and active enzyme to actually produce, much more AAT. So we don't believe that it's an enzyme or a molecule stability question. It's really that there's lower levels of RNA, and then those go up in the acute phase response, and then we can convert that to M and increase the overall amount of AAT. So that's how we believe it's working.
And as the prior question came up on the dose, pushing moving that dose to the highest third cohort since we could, is also about just looking at duration. How long? We know we've got stable construct there. So how long and then frequently do we need to administer the medicine to ensure that patients can respond?
Got it. Okay. And then I guess as a follow-up to your last comment there, Paul, what is the, residual amount of Z that you think is acceptable to support commercial uptake for Wave 006? I'm just wondering from a competitive perspective, if you need to try to push Z down as low as possible, and therefore maybe you would need to do optimization in addition to improving durability there. Thank you.
Yeah. No, it's an interesting question because as we also know that Z has a high degree of variability, right? Because as you have success on freeing up, and so I also look at this as the sensitivity measurements of being able to analyze it and it's a moment in time, but as you push up M and then therefore Z comes down, and we saw that dramatic, and it was nice to see kind of that corresponding, which is what you want to see in editing, is your Z convert to M. There's another reservoir of Z, right, which is the aggregation over liver. That's going to be will be variable over time in patient, depending on how much aggregates they have, that eventually when you substitute M, you're going to free up Z aggregates. And so it is something that we'll have to watch for over time. So again, we would expect to see, if we continue to push M higher, that Z would go lower. That's the natural, inverse relationship, with the exception of the fact that as you do these measurements, if you're clearing Z aggregates from the liver, there will be periods of time until you've completely flushed the Z aggregates out that will confound that variable to know how low is low if you're continuing to clear aggregates. And I know that seems circuitous, but you do have to look at the two reservoirs of Z. There's the hepatic reservoir, which is aggregates, and then the intracellular reservoir, which is the transcripts that are making the protein. And we'll have to follow both of those over complete time to know you've fully flushed out the lower limit of Z. But we do know that Z protein itself, and this is important, once it gets outside of the liver, has some residual function. I mean, we know there were companies that were trying to, and I think a lot of this had to do with the mechanism of doing it, to try to push Z protein out of the liver to restore some degree of functionality systemically, and those were kind of the small molecule disaggregators. So it's not that Z itself has zero biological function that's detrimental once it's freed out of the aggregates in the liver.
Maybe just add one more thing. It's an advantage both for 006 and 008 that we're modeling our medicines after human genetics. So in this case, just as a reminder, the MC patients, AAT patients, they're more or less healthy. They have very low risk of liver disease and also of lung disease. So then that means that some Z circulating is okay, right? So that's what we're trying to achieve. And the same with PLMP3, really, that, the MC or not in this case, it's like heterozygotes, they have 80% lower risk of dying from liver complications than the homozygotes. So that's the advantage of using genetics as our starting point for developing medicines.
Got it. That's very helpful. Thanks very much.
Thank you.
The final question is from Craig McLean at Wells Fargo. Please unmute yourself and begin with your question.
Hey guys, thanks for the opportunity to ask a question here. Just a quick one from us. I want to kind of go back to the safety of 007. Given the mechanism of action, how you're putting up brakes on glycolysis instead to upregulate, let's say, lipid catabolism. Can you share what you're seeing, if anything, in terms of serum glucose levels? Is this something that you've seen perhaps below a dangerous level in your patients in the study?
I'll just say the first, taking the break off lipolysis. So taking a break off lipolysis. So it's a lipolysis. So it induces lipolysis, just to make sure everybody else dial in. So that's key. But as Chris pointed out,
we really haven't seen anything like that. I mean, again, we've been dose escalating up to 600 milligrams and potentially beyond, and we've not really seen anything except for mild events. So nothing really to speak of from that perspective.
No.
And I don't think that the human data with individuals that have the mutations also would support that. So there's not really no evidence of hypoglycemia or other things even in the homozygotes. So.
Or in any of the preclinical studies that we do that would ultimately push bounds and doses as we go forward. But no, it's a great question and something, obviously you continue to follow all labs as you move into a mechanism for.
Got it. Thank you.
Thank you.
Welcome.
All right. Well, thank you everyone for joining us today. And we look forward to connecting with many of you in the very near future. Have a great day.